Okay. Is the internet clear? Am I clear now to you?
Yes.
Okay. So my topic is RNA as a target, and this is a very fast-moving, causing has been my passion I joined in the let me go through the slide here. Today Erwin Chop, and I am very delighted to present this seminar on the day. And he starts saying, on this day rather, I started saying the conviction that if different DNA species exhibited different biological activities, there should also exist chemically demonstrable differences between the deoxyribonucleic acid. And he is the one who found the Chargaff rule before James Watson and started on ISP came from Nobel laureates lab, Sidney Altman, and as well as David Epirian, John Abelson, and Tom Check, and as well as Aki Urenbeck. We were talking about nerve center of RNA research, Boulder, Colorado, and Yale University. So we trained under that pedigree of RNA research, and we brought it to India. Africa is a big continent. The wealth and the health of the nation is very important to be protected for the global order. So I present this seminar as my humble submission to the minister who presented yesterday and the chairman and the vice chairman of this conference, and as well as Professor Claudia Moraga, who is
Professor,
professor
please.
in this field.
We can't
So that
hear
I was
you. Yes, the line was breaking.
can you can you listen to me? Can you hear me?
Yes.
Hello?
Yes, please. Kindly, we apologize for any of this delay. I believe he'll be with us shortly.
unmuted it. Can you hear me now?
Yes, yes, please.
Okay, sorry, this problem is very typical to India. I apologize to you. This is internet connectivity problem. So the applications, as I was suggesting, they can be therapeutic, they can be diagnostic, they can be in gene silencing, variety of applications. I don't have to tell a lot of experts here. The particular one that I chose to work on, amaret ribozyme, it is explored for diagnostics. The way it was explored for diagnostics is because it's got a cleavage site at a substrate, and it cleaves a substrate at a particular region, and it is the cleavage site is shown there, and it is a three-dimensional structure. And all I'm saying is there are two formats of the ribozyme which we ought to know, 1, 3 and 1, 2, and I use the 1, 2, and the mechanistic activity of that amaret ribozyme is as such. And going on, the crux of the point is it is a FRET experiment, fluorescence resonance energy transfer. And we actually have put in one fluorescent probe on one side of the RNA and another fluorescent probe on another side of the RNA. So the single piece of RNA shows quenching, and when the cleavage happens, the quenching is relieved. So there is a disruption of the energy transfer. If the disruption of the energy transfer is observed, then you can easily actually monitor the real-time reaction in real time. That is the basis of the whole work. So you have quenching, you have disruption of the energy transfer, disruption of the quenching, and we can actually look at energy transfer disruption, and actually you can monitor the reaction in lifetime in real-time PCR machine like ABI 770 or 7500 or advanced machines, which I'll be showing you right now. The only point to remember here is the donor and acceptor has to be in proximity, and I will tell you in a little bit later. This is the machine I use, and all these machines are available now. So this century, this availability of the machines are plenty, and one can use, and therefore you can follow the ribozyme reaction in real time, and you can do kinetics, single kinetics, single turnover kinetics, multiple turnover kinetics. No aliquots are required. You can monitor the reaction in real time. How am I going to use that for diagnostics? Because if it is unintentional or intentional, in the case of the unintentional, I will tell you all we have to know the target and we have to design accordingly, and then we can actually do a double labeled probe that we have as a substrate, and then we have the ribozyme, and that shows cleavage. So therefore, we can follow up with the amplification methodologies like an ASBA, nucleic acid sequence detection system. This is what it is. And so nucleic acid sequence based amplification can be done, and you can detect the pathogens as such. So this can be developed as a diagnostic technique, but this is still at the bench level. There has been an Israeli company that we used to work with. It showed some promise, but this is not yet a product yet. So this is aptly suitable for African continent. And you see this G on the camera, and this side F, that is a form. There is really quenching, and this quenching you can monitor it at various concentrations of the magnesium concentration, and the double labeled RNA shows quenching. I hammered that point to you, and you can monitor the form tamra quenching is shown here. The form and the tamra, you see the peaks, and then the quenching results, and go on to show the quenching results is stabilized in the presence of the SDS and various other DMS, ethanol, and 10% ethanol. We suggested that we use that. And then here it is shown that how you can actually have a form tamra. When you put the DT tamra, that is the one, and because the guanosine is a quencher, therefore you have to put a DT tamra, and then form you should have on another end. And then we are showing the amaret ribozyme positions where you can put and where you can observe the FRET, where you cannot observe the FRET. So there are some rule available there. Then you showing how the product is cleaved and the substrate is cleaved and the products are delivered, and go on to show real-time monitoring in the amaret ribozyme. Pretty much I explained it to you. This is how you monitor the reaction. Therefore, in real time, we are able to monitor a ribozyme reaction, and therefore we can use it for a diagnostic technique. And amplified with the sequence-based detection, with the sequence-based amplification system, it has already been proven for HIV and other diseases. This can be used by Organon Technica. So we are exploring that. We are exploiting that technology to show further that the real-time monitoring can be used as a diagnostic technique. This is where we are showing 90, 30, 10, 3.3. You see the increase of reaction that is going up here. Now, I'm not going to go into the details, but let me tell you, this is another graph showing time versus the femtomoles, and it is pretty much a straight line. And therefore, here it is a group 1 intron. This is a ligation reaction. This also will be very useful. One of my colleagues did that in this lab just to show you an example. I put that in, and therefore where you exactly put the dyes and how you observe the fluorescence resonance energy transfer is a kind of important that is only known to some typically domain experts. I wouldn't pressurize that on that right now. So therefore, the same the steric hindrance to detect the steric hindrance, to avoid the steric hindrance, where do you put your dyes? That is also pretty important. And therefore, the real-time monitoring of the group 1 intron was done, amaret ribozyme was done. And I go on to show how the group 1 intron, see on the right-hand side, it shows slowly fret. And on the CBA that you are seeing is that is a quenching. That is really quenching. This is the real quenching that you are seeing here. So then if you put in the presence of SDS or urea, you can have a blood sample drawn from the wild situation, and then you can see that you can isolate the DNA sample, and or you can work with the sample of the patient, or even in the case of wild defense establishments, or people want to test that with the intentional threat, you can also typically do this and identify the pathogen. So how we are going to do that is a pretty good, interesting question. So here again, this one shows that a pretty good example of how this can be used in the presence of guanidinium and urea. And basically, we can show this real time and also analyze it by a gel. That is pretty much the example. And this is the kinetics, ribozyme kinetics that were done. I won't go into details about that, but there are some values that are given here that we can do real time. So experimental pointers are pointing out to the fact, the present work, that you can identify the RNA target, we can exploit the RNA target for a small molecule, and therefore you can use it as a therapeutic. You can as well develop a diagnostic technique. So both possibilities are there. Therefore, the artificial intelligence techniques and the databases of the RNA sequences of all the microbes, all the bacterial sequences that we have available, they can be efficiently used. It is a proprietary information. I can't reveal it to you. So one can actually find those structures common to RNA, and you can mine those structures and find where do you find these kind of stem loop structures and then bulge structures. And all these things, some of them are hidden, therefore these are inaccessible, undruggable for a small molecule, but we can find where it is druggable. As far as the diagnostics is concerned, we can actually detect an unintentional or intentional organism. So these are the works in progress. This is pretty much a deep RNA science, and nobody in India is doing it right now. But aptly can be done for African continent, who have a lot of microbial infections and as well as pathogenic infections. And India, which is infectious diseases born country, is pretty much Indo-African effort one can do, and as well as identify the target, identify the pathogen that is infecting you. This is a small molecule target. So the applications are real practical applications of a sequence specific quantitative detection of RNA directly in solution. Then couple that with the amplification techniques, one can look at the future applications. It is excellent biochemical spectroscopic physical studies. And these are some of the applications I would like to skip, but many of them are there. So therefore, it is an antisense approach is possible. The RNA as a target. We know that ribosome is a good target for many common antibiotics. So it has been explored for 50, 100 years altogether. Therefore, right now, we are looking at some countermeasures that I'm planning to propose it to defense establishments like DOD in India and as well as other countries. Therefore, we can have a machine, we can have a handheld machine, but the sensitivity is a problem to produce a handheld machine. Therefore, we can miniaturize a machine, and therefore, we can look at the technique, scientific technique to look at the intentional, unintentional pathogenic infection. Therefore, I developed this top to flow fast kinetics, and this is looking at very much an academic exercise. It is a fast kinetics, millisecond kinetics, and this gives Km, Vm, Kcat values, and one can look at all kinds of values that will be useful for commercial as well as academic interest. And therefore, the countermeasures to intentional threat, I suggested a mobile lab, because the handheld machine, there are some sensitivity and selectivity issues are there with the handheld machine. I'm not too sure. There were one company called Ibis Biosciences. They developed a T5000, which is a pretty big machine. So I don't know whether that machines can be taken to some kind of war situation or somewhere present there. But definitely a mobile lab is a real possibility. And this is a knife edge work as well as RNA science. RNA science can be a diagnostics. RNA is a diagnostic tool. Ribozyme are a diagnostic tool. And it is not cutting edge. It is not bleeding edge. It is knife edge work that Americans are buzzing about it as RNA as a drug, RNA as a small molecule market, RNA as a diagnostic. And today, if you look at RNA CAR T therapy, RNA cellular therapy, and followed by the mRNA vaccine for the COVID, this is one buzzing area all over the world. And I really, really would like the Indo-African venture to get into it and get going as far as African genomics and African Genome Conference is concerned and Indian-African collaboration is concerned. Thank you very much. I am open to any questions, but if you don't have any questions at the moment, you can always email me. That email is available with Professor Rajan, E.G. Rajan, who is also my computer science mentor. Thank you very much.